Transgenic plants. Methods of production of transgenic plants. Microprojectile bombardment, electroporation, agrobacterium tumefaciens mediated transformation. Microprojectile bombardment is a method of shooting DNA-coated tungsten or gold particles into the plant cell. Electroporation is the use of short bursts of electricity to put the DNA into cells. Agrobacterium tumefaciens is a soil bacterium responsible for crown gall disease of the dicotyledonous plant. The galls or tumors are formed at the junction between the root and the stem of infected plants. The ability of Agrobacterium tumefaciens to induce crown galls in plant is controlled by genetic information carried on Ti plasmid, tumor-inducing plasmid. Ti plasmid has two components, tDNA, transferred DNA, and the virulence gene, which are essential for the transformation of plant cells. During the transformation process, tDNA is excised from the Ti plasmid, transferred to a plant cell, and integrated into the DNA of the plant cell. The integration of the tDNA occurs at random chromosomal sites. In some cases, multiple tDNA integration events occur in the same cell. Structure of Ti plasmid. tDNA. Nopalin type Ti plasmid. tDNA is a 23,000 nucleotide pair segment. It carries 13 known genes including gene encoding enzymes that catalyze the synthesis of phytohormones, oxyindole acetic acid and the cytokine isopentyl adenosine. These phytohormones are responsible for the tumorous growth of cells in crown galls. tDNA region is bordered by 25 nucleotide pair imperfect repeats. One of these must be present in cis for tDNA excision and transfer. Virulence region. It contains the genes required for the tDNA transfer process. These genes encode the DNA processing enzymes required for excision, transfer, and integration of the tDNA segment. They are expressed at very low levels in Agrobacterium tumefaciens cells growing in soil. Exposure of the bacteria to wounded plant cells or exudates from plant cells induces enhanced levels of expression of the virulence genes. This induction process is very slow for bacteria, taking 10 to 15 hours to reach maximum levels of expression. Phenolic compounds such as acetylsyringone act as inducers of the virulence genes. This is the structure of Ti plasmid, has virulence gene and origin, then opine catabolism. tDNA region has left and right borders, auxin, cytokine, opine synthesis regions. Ti plasmid vector for creating transgenic plants. Foreign gene could be inserted into the tDNA and then transferred to the plant. In the modified Ti plasmid, the genes responsible for tumor formation are deleted. Selectable marker is added along with appropriate regulatory elements. The CAN gene from E. coli transposon TN5 has been used extensively as a selectable marker. It encodes an enzyme called neomycin phosphotransferase type 2 (NPT2). It detoxifies the kanamycin. The NPT2 coding sequence are provided with the plant promoter and plant termination and polyadenylation signals. Such constructions with prokaryotic coding sequence flanked by eukaryotic regulatory sequences are called chimeric selectable marker genes. Widely used chimeric selectable marker gene contains cauliflower mosaic virus, CAMV35S promoter, the NPT2 coding sequence, and the Ti-nopalin synthase NOS termination sequence. This chimeric gene is usually symbolized 35S NPT2 NOS. Ti vector used to transfer genes into plant have the tumor-inducing genes of the plasmid replaced with the chimeric selectable marker genes such as 35S NPT2 NOS. Limitations of the Ti plasmid vectors: production of phytohormones by transformed cells prevents them from being regenerated into mature plants. A gene encoding opine synthesis is not useful to a transgenic plant and may lower the final plant yield. Ti plasmids are larger, approximately 200 to 800 kbase pairs. Ti plasmid does not replicate in Escherichia coli, therefore it cannot be cloned in E. coli. Transfer of the tDNA, which begins from the right border, does not always end at the left border. Rather, vector DNA sequences past the left border are often transferred. Thank you.